Awesome. So again, welcome everybody to today's webinar, co-hosted by the North American 3Rs Collaborative and the IQ Consortium. This is a series on MPS webinars and workshops. And today we're going to be talking about neurological microphysiological systems. I do have a brief disclaimer to read to get us started. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussions of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither NA3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. Questions, I encourage you to submit with the new Q&A. This is actually a new feature for our version of Microsoft Teams. It's the first time using it, so hopefully it goes smooth. If you are having problems with the Q&A, you can always use the chat, and we'll answer all questions at the end of the webinar. And now I'm actually going to turn it over to Banu to give a little introduction to the IQ MPS for those who may not be familiar.
So my name is Banu. I'm an investigator in GSK, but also I'm representing IQ MPS for this webinar series. Can you give me a minute? I'm going to share my screen. So the IQ MPS in a nutshell is composed of 22 member companies. There's a good website for reference, but overall there's diverse representation from safety, ADME, the 3Rs, and PK/PD experts as well. A lot of them in the preclinical space. 2022 leadership is composed of Ree as the chair, Anya is the vice chair, and Jason Eckert now at UCB is the past chair. So each member company is provisioned two individual to serve on the steering committee. There's more than 115 active participants on several work streams that I'll get to in a minute. So what are the goals of the affiliate? There are 4 main goals of the IQ MPS affiliate. The first one, I'm just going to read from the slide so I don't misquote, is to serve as the thought leader for both MPS developers and stakeholder organizations in the industry implementation and qualification of MPS models, to provide a venue for appropriate cross-pharma collaboration and data sharing to facilitate industry implementation and qualification of the MPS models, to create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS in an industry setting. The last one, which I'm going to focus on, is develop external partnerships and collaborations to help enhance the inclusion of industry priorities. So amongst the 5 work streams, I am involved in the strategic partnerships and communication work stream here in pink. So the overall goal of the strategic partnerships and communication work stream is to communicate industry perspective and build relationships with key MPS stakeholders to enhance the inclusion of industry priorities. So in that specific work stream, some of the progress across the different years, again, I'm not going to go into detail with each of the years, but 2021 and 2022, basically beyond 2021, one of our first goals has been to establish the MOU with NA3RCs, whom we are working with now, so we can have a series of a webinar workshop combo on each organ or disease area. And now in this session, we are kind of co-partnering with NA3RCs to look at Neurological MPS webinar in a few weeks. September 21st and 23rd will be the workshop. So with that, Megan, I'll turn it back to you.
Yes, thank you, Banu. So the MOU, I saw somebody pop in the Q&A, is a memorandum of understanding that allows us and the IQ to kind of work together. So going back to my slides, that's a little bit about the IQ. The North American 3Rs Collaborative is a nonprofit whose mission is to advance science, innovation, and research animal welfare through collaborative 3Rs efforts. And actually, I realized I didn't introduce myself. My name is Dr. Megan LaFollette. I'm Program Director at the North American 3Rs Collaborative. And this is a picture of our leadership team and board of directors, and really our goal is to facilitate diverse collaborations across industry, academia, nonprofits, government, and more. And we really look for initiatives that have ample evidence, impact, and practicality. And we certainly feel that our Microphysiological Systems initiative has that. We really want to increase adoption and regulatory acceptance of MPS. Our MPS initiative is composed primarily of developers, although it does have members from other institutions. We do have an option for end users, so such as IQ members to join. So if you're interested, just reach out to me. But our primary focus is on those developers with commercially available systems. We focus on interfacing with end users, promoting regulatory acceptance, creation and update maintenance of a technology expo, and general education. So this MOU with the IQ is kind of how we interface with the end users. Yeah, so we'll be hosting a series of workshops. Right now, just in case you're new to the series, the webinars are intended to be institution agnostic and focused, at least in terms of the providers, and focused on general education. The workshops are presented by companies and actually present specific commercially available MPS systems. As Banu mentioned, we actually have our workshop scheduled in just a couple weeks on the 21st and 22nd of September. Reach out to me if you haven't been able to sign up and haven't seen that link. I can always provide it to you directly. We're also planning to have the kidney webinar on October 19th, and we'll be sending out that invitation likely tomorrow. And our kidney workshop is likely going to be on November 8th or 10th. We know 8th is election day, 10th part of the IQ MPS has a meeting, so we're still trying to figure out that one. Okay, cool. So enough of just about us. Now let's get to the real meat, what we're really excited about today, and that is our two wonderful speakers. We'll kick it off with Dr. Alysson Muotri from University of California, San Diego School of Medicine. I'm just going to pull up his bio. So Dr. Muotri is a professor of the Department of Pediatrics and Cellular and Molecular Medicine at UC San Diego. He's also the director of Stem Cell Program and Archaeolization Center. He earned his BS in Biological Science from State University of Campinas in 1995 and a PhD from Genetics in University of São Paulo, Brazil. His research focuses on brain evolution and modeling neurological diseases using human-induced pluripotent stem cells and brain organoids. He's received several awards, including the prestigious NIH Director's New Innovator Award, NARSID, Emerald Foundation Young Investigator Award, Rockstar of Innovation, NIH Eureka Award, two Telly Awards for Excellence in Science Communication, among several others. And now I will turn it over to Dr. Alysson Muotri.
Hi, thank you so much. And I'll get started by sharing my slide here. Yeah,
Great.
Thanks, Megan, for the introduction and for the invitation. I'm hoping you can now see my slides.
Yes, they look great. Thank you, Alysson.
All right, fantastic. So what I decided to do is to give an overview of the use of brain organoids in my lab and a little bit of the technology development, passing through some of the history of the technology and the major roadblocks that we have until we fully adopted these as a replacement, for example. And then I'll end with some potential applications, just a hint on that. I know that Stefan might take over and go more in deep into that, so I'm not gonna show any full example, but just a hint on potential applications that we're taking here in the lab. So part of the reason why we're using brain organoids is really to study neurological conditions that have oranges, genetic oranges in utero during the embryogenesis, and this is hardly accessible to investigators. I mean, I'm showing here an example of an ultrasound, so you don't have the resolution to see what happens at the synaptic level, at the neuronal level. You cannot record from neurons at that stage. So we still don't have no invasive technologies to study how the brains form. So the use of brain organoid allow us to recapitulate human neurodevelopment outside the uterus in a controlled environment of the lab. And that's why I was very excited about this technology. And this was exactly when I was setting up my lab. The brain organoid technology was initially pioneered by Yoshiki Sasai. That was in 2008. So in this paper, he actually shows that if you neuralize human embryonic and human iPS cells in a three-dimensional condition, the cells will self-organize and start forming these tissues that have a remarkable similarities to the human fetal brain. So here are some of his original pictures. You can see like the formation of the cortical plate, some of the rosettes. It was actually amazing. I remember reading that and said, well, that's what I want to do in my lab. And so we decided to start working on that. But as any new technology, I mean, the early days were quite hard. So much easier to make two-dimensional neurons in culture than really to recreate a piece of a three-dimensional brain. But nonetheless, I mean, eventually we and others, we all catch up with this technology. And now I would say that is a much easier way to do it. And many people have evolved these protocols over the last 5-10 years. And I'm showing here just a summary of the protocols that it's out there. For example, you can have unguided differentiation, stimulating the formation of different brain parts in the same unit. So this is actually really cool because you can have, for example, a piece of the retina, a piece of the cortex, some hippocampal cells in there. The bad news is that this is disorganized, and each one of them will develop on its own. So there's no control here. So it's hard for disease modeling, but it's good if you want a representation of the different brain parts. On the opposite, you can add ectopic factors in these cultures and induce a regionally specific organoid. For example, you can add a dual SMAD inhibitors and drive these progenitor cells towards the cortex. So you have cortical organoids. And moreover, I mean, you can create different brain pieces and fuse them together to create what we now call an assembloid. These organoids, they spontaneously fuse to each other, and in some instances, they'll make the connections. It's very far from the circuitry that you have in the brain, but it is a start. So there is advantage and disadvantage on these protocols. It really depends on what you want. You might choose one versus the other. But it's nice that there is room for improvement. So we can create other things, and people in the lab are constantly improving the protocols to become more robust, more reliable, and more functional. And that's more or less what I'll push here on the talk, how to create this functional organoid. But before I proceed, let me just say that we are not yet on the stage where we have like a mini brain a dish, and this is most of the time what the media will pick it up and call mini brain because it's a direct contact with the reader, but gives you the impression that you have the intact tissue in a petri dish, and that's not the case. There's actually lots of limitations that we still have to overcome. These cells are quite immature. We don't have vascularization. Not all cell types are represented, and even the conditions that we grow these cells were not being optimized. So it's hard to say how translational are these technology. But again, we have like good evidence now that things are improving and we are getting better and better. My lab, personally, what we like to do is to take advantage of DOE. It's called the design of experiments. This is a century-old mathematical model. So biologists usually they avoid this kind of modeling, but it's quite useful. So it allows you to optimize the interaction between different factors in a specific protocol. For example, the concentration of FGF and the pH. So you can systematically create these matrices to optimize all these different factors, remove what's neutral, adjust what is negative, and improve what's positive. So you fully optimize the conditions, and we did that to grow organoids that have more healthy human neurons, so they would fire. Because before that, really, we have spontaneous activity in neurons here and then, but nothing compared to the real thing. So long story short, it took like 5 years to optimize the protocol for cortical brain organoid. We grow them in conditions that are fully chemically defined. It's an ideal media. These are single cells. We neuralize them with FGF followed by EGF. And then we start the differentiation by adding the transient exposure to the dual SMAD inhibitors. And after a while, we just let the cells go. So we are moving from single cells to structures that contains about 5 million cells inside. And they can grow up to 0.5 centimeter in diameter. And if you've never seen one of those structures, this is Isabel in the lab, and you can see a plate here. Each one of these white balls, you can actually see them at naked eye, is one of these 5 million balls of organoids. It's the equivalent of a bee brain that you have in there. And they self-organize in a very similar way as the human brain does. What you're seeing here is a video or a two-dimensional representation of what happens inside the organoid. You have lots of neuroprogenitor cells here on top. These progenitor cells migrate out from inside to the outside of the organoid. They use radioglia cells to guide themselves and form the cortical plate. So this inside out, it's what happens in most of in all the mammalians' brains. And as the cells mature, they start to form the connections, and that's when we start to see the electrical activity. This is how they look like. If you cross-section one of these organoids, you have a rosette full of neuroprogenitor cells surrounding this ventricular zone. Here in white, you have these radioglia cells guiding the production of the neuroprogenitor cells that will end up forming the cortical plate. As the organoids mature, the diversification of neurons in the cortical plate increase as well, together with the electrical activity that I'll show you in a bit. Most people ask me what kind of cells you have in there. So I told you that we start from single cells. We end up with 5 million cells. What is in between? Well, it's very dynamic. It depends on what stage you are looking at. I'm showing here a snapshot at 4-month-old organoid using single-cell RNA-seq. You can clearly see that most of the neurons that we have in there are glutamatergic neurons. You already start to see a division between upper cortical layer and lower cortical layer. There's a pool of progenitor cells, transient progenitor cells that will start to become glia cells. There are the radioglia cells, as I mentioned, but at this stage, we start to see the proliferation of astrocytes. At that stage, we also see some of the GABAergic markers being expressed. It's interesting that those neurons are not yet inhibitory neurons. Inhibitory neurons will take a while to appear, but the markers are already there. So they'll evolve into fully functional inhibitory neurons a little bit down the road, but they already show the markers at that stage. You can also infuse the cells to create assembloids with different cell types that do not spontaneously emerge into these cortical organoids. So here we create microglia-like cells and we infuse them inside the organoids. Here in red are the microglia cells. They like to live inside the organoids and they start to proliferate and stay as a resident immune system inside these organoids. So you can actually create immune-responsive brain organoid on that way. All right, but I started by saying that I want to model conditions, neurogenetic conditions that start in uterus, and these are autism, schizophrenia, things like that, and they all require a defect in networks, and those original protocols of brain organoids didn't really provide strong networks. They barely provide functional neurons. But after the optimization, we decided to check if they are functional in our hands. So we use these voltage-dependent dyes that you can just add to the cultures, and you see the function of the neurons. You can label some of these neurons, so you see these beautiful pyramidal cortical neurons full of spine. So this is a sign of maturity. We like to patch clump those neurons as well, and we see that over time they actually increase excitability. This is exactly what we'd expect for a neuronal maturation. We can do multi-auto patch by patching 100 neurons at the same time. So we start to get like a hint of the maturation of these cells. There is one thing that was missing was some kind of ensemble of these neuronal networks. For example, I'm giving like two ideas here. If you take the organoid as an analogy for a full stadium, if you're just patching a couple of neurons, you are losing, for example, the OLA behavior here, or what happens here that everybody was silent and then suddenly everybody's excited. So we need methods to kind of visualize all these entire populations of the same time. And honestly, one of the few technologies that allow us to do that is the multi-electrode arrays. So we start by plating the organoids on top of the electrodes, and over time we capture the electrical activity, so we can measure the excitability of the entire network using this technology, and that has been proven to be very user-friendly for us. And at the beginning, I must say that I was very skeptical that this would work or that we would see anything different, mostly because previous attempts to record from multi-electrode arrays on 2D cultures never really could pass the 5 Hertz threshold. That's what I call very immature neuronal-like network. And the primate brain will fire around 20 Hertz. A mouse brain fire around 18 Hertz. So you can see that how our 2D mouse rat cultures are really not up to representation of the in vivo network. This is from my colleague, Paul Arlota, who actually put the electrodes on organoids up to 32 weeks. And as you can see, they cannot cross the 5 Hertz threshold. So that's why I was not super excited. I didn't think that optimization of the protocol would lead to anything different. But fortunately, I was wrong. And I was wrong in a big way. Here in red, you can see the increasing in mean firing rate every week from an average of 8 organoids. And after 25 weeks, if you remember, that's when the astrocytes start to kick in. You start to see almost exponential activity of synapses, leading to an increasing mean firing rate that reaches this 20 Hertz when the organoids are about 40 weeks or 9 months of age. So as long as you keep the organoids in these optimized conditions, you give the right environment for the neurons to thrive, to make connections, and become highly active. So this was very unusual. We are moving from 3,000 spikes per minute to something that is 300,000 spikes per minute, which is way closer to an in vivo neuron than what we used to have in tissue culture. I'm just saying that how unprecedented this is in vitro. We never had like a system that would evolve in that way. And we have both excitation and inhibition, so you can actually detect most of the neurotransmitters using mass spec. Just this representation of the GABA that we start to see the release of GABA exactly when we see the release of GABA in human fetal brain, and you can block the activity of glutamatergic neurons, and you see that the number of bursts in neurons also reduced. So this is to say that we have both excitation and inhibition in those cultures, and they are working as expected. So when we first had that data, I was showing to a friend of mine, Brad Wojtek here at UCSD, and he said, wow, I mean, if you do have that much activity, these networks in the cortex must start generating neural oscillations. For those who are not familiar, these are brain waves that you can capture using electrodes throughout your skull, and they change upon human activities, and this is what cognitive neurosciences have been doing for a while to correlate the different types of neural oscillations with human cognition. For example, if you have a seizures, you're going to see that those brain waves will dramatically change. If you have schizophrenia, something, autism, the same thing. If you're paying attention or sleeping, your brain waves will change. So the ability to correlate neural oscillations with cognition is something really powerful. But we, on the fundamental side of biology, we are still looking at synapses, cell types, things like that, and we're never able to connect those things with human cognition. So we thought that, well, I mean, if we do see the brain oscillations in the organoids, for the first time, we might be able to bridge the fundamental biology towards human cognition, and that would be really, really cool. So we team up with him to look for these neural oscillations, and we develop a protocol to take advantage of the spike raster plots from the multilateral arrays. We create the population spiking and the local field potential that is the proxy for the detection of these neural oscillations. And then we ask, can we actually see these oscillations in the organoids? And the answer is yes, but you have to wait at least 4 months. So in the beginning, we don't see many oscillations. The evidence of oscillations starts together with the emergency of astrocytes, when you see these second peaks rising from the organoids. By 6 months, it's very clear, very stereotyped. You have lots of oscillations. And by 8 months, they become highly complex. This is what you would expect for the cortex. This complexity is related to the emergency of the inhibitory neurons, or the functionality of the inhibitory neurons. That's when the inhibitory neurons actually become inhibitory neurons, and they start to evolve these neural oscillations to something as complex as different frequencies that we see in the cortex. So we, over time, over the course of 9 months, which is a human gestation time, we are moving from very rigid network to something that is complex, flexible, abundant, and recurrent. And that's why we see this inverted U shape on the number of sub-peaks of these oscillations, because they start to overlap. So there is a peak around 25 weeks, and then they start to overlap a lot. But that's a sign, again, of a highly complex network. So we're very excited about that, and we start talking about this data to others, and we send the paper to a journal, and we got the same questions all the time. That's great that you see these oscillations, but how similar are they with the human brain? And that's a difficult question, because as I told you, we don't have access to recording of networks inside the uterus. How can we measure what happens inside the uterus? So we decided to rely on preterm baby's brain, EEGs. So when babies are born prematurely, we can place the caps on their heads and just record from them. That's what we call the preterm neonatal EEG. And as you can see here, this is just a representation of a typical neonatal EEG. It's composed by spontaneous activity, transient spontaneous activity, followed by quiescent time, or where the brain seems to be resting. And it's very different from an adult EEG, where there is no resting anymore, because everything is already fully connected. And if you look at the organoid, the LFP resemble this preterm neonatal EEG, which is what we would expect for the normal neurodevelopment. But the question is, how similar is that? And so we decided to take advantage of artificial intelligence. We create a machine learning organoid where we train the machine to detect the age of the subject based on its EEG. So these are babies that were born between 25 and 38 weeks. And we have over 500 EEGs, and the machine gets really good. It tells the age of the baby, the correct age of the baby, just by looking at 2 or 3 EEG features. So we use this regression model now to feed with the organoid data. This is the data coming from the multilateral array in a very unbiased way. So the machine doesn't know if it's looking to the human brain or if it's looking to the organoid. And we did that with organoids over the span of 9, 10 weeks. And then, I mean, we asked the machine, can you guess the age of the organoid? And the data was incredibly, the machine gets really confused. It cannot distinguish between the organoid and the human data. And this is what I mean. I'm plotting here the predicted age versus the actual age. On the dotted line is the perfect correlation. The black dots are the data from the human EEG, and the blue dots are the data from the organoid. You can see that it's more variable, but it follows exactly the same trajectory, and this is highly significant. The correlation is very poor before 25 weeks. The reason is we don't know what happens before 25 weeks. We never train the machine with EEGs on that early of a brain. We don't have the data. This is still a black box. Perhaps we can use the organoid to guess how these networks emerge, but we have so far no way to analyze that. It's an ethical roadblock that we have, so we cannot do that with the current technology. All right. So now that we have these EEGs, oh, this is just one example of, for example, the inter-event interval, meaning that as you grow older, the silent period between these two specific activity gets shorter and shorter, giving rise to the adult EEG. By the way, in your life, you have this stereotype EEG when you are born, and just minutes after you die. So all the rest of your life, you have like a full EEG, like this one here in the bottom. So the organoid follows the same trend. It has these EEG, the intervals become shorter and shorter, reaching the same level of a newborn baby brain. So that was quite impressive for an organoid with only 5 million neurons, but they can do it. So it's not about neuronal numbers, it's not about brain size. It's all about connectivity that you can see in those neurons, which I think is really good news for this field. So where are we now? We want to continue to improve the organoid technology by stimulating these organoids. So we feel that when you are born, one thing that happens is you become aware, your senses become aware. And one of the first things that happens is that you open your eyes and you start receiving the visual stimulus, among other things. I mean, you will learn how to walk as well and develop your motor cortex. But opening the eyes is something that we think is quite important for the brain. So we are teaming up with Carl Walling and Chao Chen to create this circuitry where we have the retina organoid fused with the thalamic organoid, fused with the cortex organoids. So you can visualize the photoreceptors using reporters, and we can visualize the projection, the axonal projections of the retina, innervating the cortex using different reporters as well. So we have those structures. They seem to fuse really well. They're maturing now. This is just a recent picture of the retina organoid here in the bottom, and you can see it passes through the thalamus and is reaching the cortex, the cortical organoids really well. So this is it's amazing that it can do it so well. So we actually pass through this potential optic channel here and innervate the cortex. So we are waiting for these photoreceptors to mature so we can stimulate with light, and then we can record from the cortex, and we can ask questions is, can we increase the maturation? Do we see different oscillations? Do we have now a visual cortex instead of an empty canvas? Things like that. So, I mean, we allow us to really understand how the stimulus, the input and output will play a role on cortical development. So the other idea, it is to stimulate the organoids using an entire body, right? I mean, because that's what happens in vivo. Your brain is not connected from the body; it's actually fully connected to a body. And people were thinking about that. Some labs are, including ours, are transplanting brain organoids inside animals. But I don't like this approach. I think that now we have a chimera that doesn't really fit the purpose. I think it's okay for a proof of principle, but it's not high throughput, and we still have the blood of the animal passing through. All the vascularization is coming from the host. I don't think it's an ideal system. So we thought about something different. Well, I mean, if we cannot yet mimic the whole body in a dish, perhaps we should take advantage of something that's artificial. So we team up with CENI. This is the Center for Engineered Natural Intelligence here at UCSD. I'm a faculty there as well. And we decided to create a brain-machine interface to be connected with the organoids. So what you have was something that was inspired by me watching like a Star Trek movie where these very old monks who achieved the kind of nirvana they disconnected the brain from their bodies, and they are now in that state where they don't feel the pains of the human body, so they can be much more elevated in a way. So we thought that, well, I mean, that's the kind of thing that we need for the brain organoid. We need a machine like that. And what you are watching here, it is our attempt to teach a robot how to walk using these neural oscillations that are coming from a brain organoid that are sitting inside an incubator and sending the electrical signals to the robot via wireless. We are now adding sensors to the robot. You can see those eyes. These are infrared sensors. As the robot gets closer to an obstacle, it starts to stimulate back the organoid inside the incubator. That stimulus changes how the network is rearranged by creating different types of circuitry inside the organoid. We have now a computer that detects when that happens and send, this is the peak of stimulation as the organoid reaches the wall. So it changes the network, and then we have a second command to the robot, such as stop and walk back. So we are repeating this experiment over and over, including a specific maze where the robot has to avoid the different obstacles to achieve a specific task. And our goal is to learn how the human brain learns. But we also are going to learn how these different stimulus that happens throughout the environment can actually accelerate or not the maturation of these organoids that are just now sitting inside the incubator. So finally, I must say, and I'm pretty sure that Stefan will cover that, we also have applications for biomedical purposes, and this is the bread and butter of the lab. These are some of the conditions, the genetic disorders that we now have models, sometimes isogenic, sometimes patient-derived models, where we create iPSCs and then we generate brain organoids. We use that for drug testing, gene replacement, gene editing. Some of these conditions we don't have good animal models, or the animal models really suck. So we hope that in the future this type of technology will allow us to move faster towards clinical trial. We have like several examples where we take the drugs from early stages and we're already like in clinical trials. Others we are still trying to understand what are the genes that are implicated. So different stages for different diseases right now in the lab. All right, so I'll stop here with my take-home messages. And hope that I convince you guys that there is still room for improvement on this technology. Brain organoids can be derived from both embryonic as well as induced pluripotent stem cells, and can be made in several different ways, in several different brain regions. It's not perfect, but we're getting there. They have similarities at the molecular, cellular, and anatomical organization with the human fetal brain. I think many labs have seen that. I think there is no question that this actually happens. I'm glad that this is something intrinsic of the organoids. It's not that we have to do it. They have all the genetic information encoded in their genome to produce these structures. Finally, I think we are seeing improvement on the electrophysiological signatures of these organoids that mimics the development of the human brain, and I think that's huge. This is something that we never had before. For the first time, we can actually follow those network development. So it's a huge step for conditions such as autism, schizophrenia, dementia, things like that. There are several uses for these brain organoids. I point to some of them. Understanding human development. We now have a huge emphasis on brain evolution, trying to understand why the human brain is so unique. Disease modeling, as I said, drug discovery, toxicology, and we hope that this technology would be more and more accepted by the FDA and other agencies as a replacement for mouse models or anything related to non-human models. So if I have to put all my money, I'll put my money on improving this technology to make it closer to the real thing, rather than going back to animal models. So that's where we are. and I didn't have the opportunity, but this work was done by many members of my lab, so I didn't highlight anyone specifically. So I was not alone doing all of that. So thank you so much, and I'll stop sharing here.
Awesome. Thank you, Alysson. So just a reminder, everybody can put any questions that they have in the chat, and I'll invite Stefan to go ahead and turn on video and microphone and share screen.
Yes, thanks. So let's try to share my screen.
Yeah.
Should work.
Loading. Yeah. Okay, looks great. Thank you. Oh, and I'll read Stefan's bio real quick. So Dr. Stefan Kusterman is a principal scientist at Investigative and Immunosafety in Pharmaceutical Sciences. Together with the team of I2S, he is responsible for in vitro safety assessment of new drug candidates by using cellular in vitro models, mainly in the areas of brain, eye, and teratogenicity, as well as developing new models and strategies to improve safety assessment. So take it away, Dr. Kusterman.
Thank you very much for the kind introduction. And thank you for kicking this off with such an exciting talk. I'm not going to show any robos. I will give you a bit of an industry perspective of use of these MPS models, specifically in the preclinical drug development, and here with a focus, as I'm part of the preclinical safety assessment, with a focus on preclinical safety. Before touching on the models, I would like to kick off my presentation with some considerations why over the past years in pharmaceutical industry, the application and the development of such complex models have been so much in focus. So what are the main reasons? And one of those reasons are here, if you look on the graph on the left side, is a development and a shift of our portfolio with regard to therapeutic modalities, where in the past we had a very much small molecule dominated portfolio, and today there is a variety of new modalities, being large molecules, RNA therapeutics, gene therapy, you name it, which make more or less up to 50% of the portfolio. And this shift in the portfolio triggers also the need for a shift and a change in safety assessment. And the reason for that are partially because these new modalities are complex molecules, as I said already, and some of them, they are so human specific that they do not cross-react with any preclinical species. And we need to have some cross-reactivity to make sure that we can capture all the different aspects of safety. So for that, sometimes preclinical in vivo testing may either not be relevant or is even unethical, because there's no reason for running animal studies for preclinical safety, specifically also in oncology. So, and in addition to that, the target space has also shifted, and at least my subjective view on that is that the targets became much more complex, and the mechanism of action we are trying to target is quite getting more and more complex, and there is a need for an improved mechanistic understanding of the targets, and this also with the aspect of toxicity and safety testing. And lastly, of course, there's also from the outside a society ethical concerns which really question the use of animals, even for safety assessment or testing of pharmaceuticals, how we do it today. So this taking together started that we really looked into investing into these models and pioneering these fields also at Roche, and make sure that we can capture some of these points. So today I would like to, actually we haven't aligned on that, but I would like to capture on two model systems, which is on CNS toxicity and in addition also retina toxicity. Retina being part of the CNS also fits very well into the neuro MPS we are covering today. So I would like to kick off with the retinal toxicity. First of all, I mean, if we talk about the eye, it's kind of obvious that it's quite a complex organ. And if you look into the histomorphology of the retina with its different cell types being neurons, glia, and pigment epithelial cells, which all have their specific function, it's quite hard to model this in an in vitro model. However, there is a bunch of reports showing drug-induced toxicity of the retina, which could, I have just depicted it here from a recent publication, that this could trigger RPE malfunction, photoreceptor damage, ganglion cell optic nerve damage, and so on. And this, of course, leads to some quite devastating toxicity in patients, ultimately leading to blindness. So, for example, systemically applied drugs, one of the very often used controls also for in vitro model development is chloroquine, which is known to induce after a certain dose and time could lead to bullseye maculopathy, which could then in the end also destroy photoreceptor cells and subsequent neurons, and could lead to an impaired vision. But then there's also other drugs, more recent developments in the field of IVT applied drugs like MAPS, and also gene therapy, and they are known to induce immunogenicity or immune-related effects, which could again lead to some impaired vision. So taking this together, there is a need to better understand these sorts of toxicity and also have some tools at hand to early check whether some of our drug candidates early in the pipeline would lead to such toxicities. So if you think about retina in vitro models, there's obviously two choices. So number one is there's retinal explants. This is, I don't know for how many decades such a technology is available. So it's actually, it's explanting the retina into an in vitro dish, as you can see here. And they all come with their advantages and challenges here. Depending on the protocol, the tissue preservation over time is quite high, as you can also see here. After 42 days in vitro, still photoreceptors, including some shorter outer segments, are well preserved, the synaptic pedicles here. So overall morphology looks quite good. Enables, of course, the use of patient material, which is then also a limitation on the other side, because the tissue availability is really sparse and is very limited. And the lack of light sensitivity over time. So the sensitivity of those retinal explants, again, depending on the protocol to detect light and measure it by electrophysiology, is decreasing quite rapidly. And of course, there is no vascularization, which plays a major role for modeling many diseases of the retina. Another model are now more recent one is retinal organoids, like brain organoids. They are iPS-derived organoids. They also display a lot of different cell types of the retina. They also display, to some extent at least, the morphological layering of the retina. Enables again the use of patient material, which is of interest if we talk about the monogenetic diseases, for example, of the retina. Is a scalable model, no need for tissue donors. However, as for some other of the iPS-derived models, they come with the disadvantage of the immaturity of the model. They lack a photoreceptor RPE interaction, which is crucial for the phototransduction cascade, at least most of these models, and they again lack a vascularization. So we started looking into the retinal organoids as a potential model and developed a lifestyle imaging-based readout to capture retinal toxicity. And we looked, or we started doing that by a lifestyle imaging-based approach, looking into apoptosis and necrosis in a lifestyle imaging fashion using an Incusight device. And you can nicely see by applying a couple of well-described neurotoxic or general toxic compounds, you can nicely see that that you get a dose-dependent increase in apoptosis, and for some also into the necrosis feature. We compared it to some very classical standard readouts usually used in vitro toxicology. The one is LDH release and ATP content, which measures the destruction of cells, which then lead to a release of the LDH, the intracellular content into the supernatant, and can be measured. And also ATP, which gives you a measure for overall viability of the organoids. And what we could find out is that this lifestyle imaging readout, which is much more straightforward, nicely reflects or recapitulates our ranking we have also observed with the 2 standard readouts. So our POC study with the tool compounds demonstrates the feasibility to detect apoptosis and necrosis to some extent in the system, and the data correlates well with some very standard readouts we usually run in vitro toxicology assessment here at Roche. So from that we went on, and we wanted to screen AAV constructs in our retinal organoids, and we applied the same methodology. Again, we focused here on the apoptosis readout, and we compared it, our AAV constructs, which are down there, with the different positive controls for toxicity. And what you can nicely see after 14 days of incubation, you see that the AAV constructs, they do not induce any toxic effects, which is apoptosis, caspase-3 cleavage, compared to the positive controls. We went further on. This was 21 days after transduction, and we measured for 14 days. So we went on and we did some single-cell RNA sequencing, and we looked into some markers, gene markers of unfolded protein response, necroptosis and apoptosis in the most important cell populations, which is the photoreceptors, rods and cones, and also the Müller glia cells. And what we found out is that none of our constructs did induce any increased expression of neither of the markers, being UPR, necroptosis, or apoptosis, which nicely matches with our previous data, where we couldn't see any caspase-3 activation. So this is one example how we have applied the retinal organoids to screen early on for potential cytotoxicity induction or retinal toxicity induction by AAV constructs. However, I said at the beginning that one limitation of most of the retinal organoid models which are published so far is that there is a lack of RPE photoreceptor interaction, which is quite crucial because here the phototransduction cascade takes place, and so this is an important process in physiology of the retina. So together with our collaboration partners at the University of Tübingen, we started looking into options how we can tackle that, and one of the solutions they came up is combining the retinal organoids with a microphysiological device in a way that you seed the retinal organoids on a microfluidic chips where they sit on a monolayer of RPE as they would do in vivo, and by that enabling the interaction of the photoreceptors with the RPE. So another advantage of enabling RPE photoreceptor interaction is also that we could mimic in such a microfluidic device the main application routes for ocular drugs, which is either intravitreal injection. All the large molecule drugs for wet AMD, for example, are injected today via a syringe into the eye. So that would be the intravitreal exposure route, or some of the drugs are applied by systemic exposure as chloroquine, which is not an ocular drug, but it induces toxicity in the retina. So we could mimic both routes of application and administration here by injecting either the drug into the gel, which then exposes the retinal organoids, or we just add the drug into the flow. We wanted to test that as said, it's a vascular-like perfusion, enables photoreceptor RPE interaction, and enables the systemic versus IVT drug application to be mimicked in this system. So a quick word on this. So the photoreceptor RPE interaction, as said, is quite crucial for retinal physiology. And this here recapitulates the in vivo outer segment recycling process, which also takes place in vivo. And here the photoreceptors are labeled with PNA lectin, and the retinal pigment epithelium is labeled in green. And you can see already after 1 day on a chip that some of the red labeled outer segments of the photoreceptors get in quite a close proximity to the green labeled RPE. If you look after 7 days, these outer segment processes, you can also find them intracellularly in RPE cells, which is here shown by electron microscopy. And after 21 days, you see a strong interaction between the recorrine labeled photoreceptors, and you see the recorrine staining is also present in lysosomes here of the photoreceptors. So this gives you some evidence that indeed photoreceptor outer segments are shattered by photoreceptors as done in vivo, and they are taken up actively by the RPE cells, which is their task also in vivo. So we tested this systemic versus IVT application of drugs in the chip, and we applied the well-described chloroquine control to our retinal organoids on a chip. And what you can see is, as expected, after a dose-dependent increase in toxicity after 3 days with a very high dose of chloroquine, and also after 21 days with lower doses, you get an increase in toxicity, as here shown by propylium iodide staining. However, we also wanted to show that that one of the causes of this toxicity is going to be captured in our system, and this is a lysosomal function / dysfunction, which you can see nicely that you get a lysosomal swelling and accumulation, which is shown here by an increase in the staining of LAMP2 in the system, which shows that indeed the process, which takes also place in vivo and is part of the retinal toxicity mechanism of chloroquine, which is supposed, is also recapitulated in the retina on a chip system by applying the drug. As said, it's not injected into the retinal organoids, but is simply added to the microphysiological flow, where all the nutrition of the retina and RPE takes place. So no additional chloroquine is added. So this can be nicely mimicked in this retina on a chip. In a next step, we also wanted to test this for an IVT-like application with some internal compounds, and to ease the penetration of the compounds into the retinal organoid and make the structure more in vivo-like, because in vivo the retina is also not a closed ball, but rather an open half ball, if you want. We started to cut open the organoids and make like a half a globe, which you can also see here in this microscopy image, 3D reconstruction. It's the PNA lectin again, stained retinal organoid, which sits here on the RPE monolayer in the chip. We applied some compounds into this, and what we can see here in different concentrations after 28 days after one single application of the drug, you can see that that there is a nice increase in these in the toxicity stain of the retinal organoid, but not of the RPE. And this exactly was recapitulating what we also have been observing in previous in vivo studies, and which is quite nicely recapitulating the overall in vivo phenotype, which for us makes it a useful system to test such compounds also in this model with an IVT-like application. So what's next? One important factor, not only in the brain, but also in the retina is, of course, the immune system. And as said, we have Müller cells, the radial glia of the retina is present, but also microglia cells play an important role. And together, again, with our collaboration partners, we started looking into the integration of the microglia into the retinal organoids. And what you can nicely see here, after 7 and after 14 days, M-cherry labeled microglia cells, they nicely settle down in the retinal organoid. And what is even nicer, in my opinion, is that we could also make a proof that adding an injury to the retinal organoid also leads to a physiological response of the microglia, which is here a laser injury has been set to the retinal organoid, and after 10 minutes you can, these are the red labeled microglial cells, and after 10 minutes you can nicely see that the labeled microglia cells, they start migrating to the injury point and start fixing it as they would also do it in an in vivo situation. So this is ongoing, and I think that's an important step also towards best recapitulation of potential toxicities in such a model system. So in summary, retinal organoids are useful models for preclinical safety testing, which I have been demonstrating with some examples. On a chip design even enables on top the RP photoreceptor interplay, which is a crucial physiological function and also important for disease modeling. The model enables 2 application routes, which is also for toxicity and PK/PD analysis could be of importance, which is IVT versus the venous application. And then we have microglia, which can be integrated into the organoids and responds to injury in a physiological manner. But however, there is also still some room for improvement. Of course, the flat morphology and the barriers, which are also important for drug penetration into the retina and better modeling these features of different drug candidates into the retina, are not entirely recapitulated. For example, inner limiting membranes, outer limiting membranes. The retinal organoids are more resembling immature tissue, as is also true for brain organoids. And there is still some room for improvement for batch-to-batch reproducibility and having these models more homogeneous and to make the individual experiments more reproducible. So maybe automation is one factor which could do that, and maybe others. This is still work to be done and partially ongoing. So far for the retina. Now we switch gears for CNS toxicity. So if you think about the brain, I mean, it has nicely been captured already. It's such a complex system that we have to capture a lot of different things if we also think about toxicity in particular. So we have been starting such a project by having been approached that we have we wanted to investigate human relevance of a neurotoxicity we observed in a non-human primate study. And we started modeling this in a 2D iPSC-derived neuroastrocyte co-culture model for 14 days. And what you can see when, again, looking into apoptosis, you see an increase of apoptosis by some Roche internal compounds in the cyno model, but not in the human model. And that, for us, was kind of the trigger point to invest further into that and render these models more relevant and look into 3D models for that. So we started looking into cyno 3D brain models, a spheroid protocol by applying cyno iPS cells and let them grow into spheroids over a couple of weeks. And what you can see nicely that after 4 to 8 weeks, we have a nice presence of neurons, astrocytes, oligodendrocytes, and even myelin we could show. This also was confirmed by gene expression of different marker genes of astrocytes, oligodendrocytes, and in the progenitor and the mature stage. We went on and we also did the same for the human brain model, and we could also identify that that in the human brain model we have different cells present, as already has been shown by the previous speaker, and this was again confirmed by marker genes. One aspect which was quite important for us was a proper myelination, and this is what you can see here. Again, 16 weeks, we see MBP GFP nicely outlines neuronal processes, and here also in a 3D rendering, the NFH labeled neuronal processes are nicely enwrapped, some of them by the green MBP GFP. And in addition, as said for the retina, also the microglia is an important component. We also did work on integrating microglia into this brain spheroid model. And the microglia did integrate well into the human spheroids and remain stable for at least 4 weeks within the brain spheroids. It shows a ramified structure, which is indicative of a more homeostatic phenotype. We also then finally did look into an imaging-based workflow to assess CNS toxicity, again applying the caspase-3/7 activation assay. Here again with some positive controls, you can see a nice dose response of the MPP+, for example, by that imaging device. And we also did quantification of this finally, and we could show that MPP+, rotenone, and also for nefazodone, we can recapitulate some of the toxicities which we would like to show. So taking this together, we have shown that the model demonstrates a physiological relevance with the major neuronal cell types present. The integration of microglia and autologous integration is possible, showing a homeostatic phenotype and a long survival within about more than 4 weeks. Cyno-iPSC-derived cerebral spheroid model shows high physiological relevance as well, because it has is quite comparable to the human model, and we can quantify neurotoxicity in these 3D models. However, the spheroid model does not resemble, of course, in vivo morphology of the brain, which could be important, maybe not so much for the toxicity, but for other aspects. An iPS-derived model is still considered immature, which is maybe for toxicity assessment, it is less sensitive. And BBB, as a more expanded step, is not part of the model, which might not be a true limitation for some of the things for toxicity assessment. So as an outlook, maybe now the complex models for brain and retina are available or in a real advanced development stage. Ongoing is really to further demonstrate the relevance of these in vitro models for the drug development by continuous generation of use cases and publications for this preclinical drug development. And of course, we still need to broaden the application of the complex cellular models within the pharma research and development, also for disease modeling, gain further acceptance in the field by scientific, which is the scientific communities, regulators, by sharing knowledge, openly publishing successes, but also their limitations. And then overall, of course, an aim is to replace the animal testing to increase human relevance of drug safety assessment. So that is not all my own work, in a sense of most work has been done by the people in the lab, which is in ISAF is Christine, Kevin, Michael, and Sabine, then also in PS, in the entire Roche family, and also our collaborators here in Tübingen, who did a lot of work. So thank you for your attendance, and now I guess happy to take questions.
Awesome. Thank you, Stefan. If you want to stop sharing your screen and Alysson to come back on as well. Awesome. Welcome both. Thank you both for the talks today. Reminder to please put any questions in the Q&A. I'll go ahead and start off with a relatively general one. So how are or should neurological MPS systems be validated for use, either for assessing neurotoxicity or drug efficacy? What are some important factors for validation? And either take that question first.
I want to go ahead, Stefan.
From a company perspective, I guess, I mean, let's answer it differently. I think one of the challenges in toxicity assessment is, of course, you never know what toxicity to be expected with the next modality, with the next compound you add. So therefore, we think a lot of these things, I mean, disease modeling and the better understanding of the disease is, I wouldn't say it's a low-hanging fruit, but it's maybe a lower-hanging fruit, or is a good starting point to continue this journey. And then in these models, which have been then developed specifically for the disease modeling, then also the compounds can be tested as an adjunct assay, so to say, using some of the technologies I have been showing. So I think disease modeling is an important aspect also with regard to animal usage and so on. And then the toxicity can kind of follow these routes.
Yeah, I would add that there are certain toxicity that we can already use the current technology, and we are doing that, for example, for gene therapy. There are certain genes that overexpression of those genes might be important, and it's definitely like a safety issue. How can we be sure that we control the right levels of expression upon a gene therapy in a human cell? So the really only way to do that is by infecting the human target cell in vitro and see if you maintain the same level of expression. You can also see if you overexpress that gene in the cell, what kind of consequences you have. And neuronal cells are very flexible to gene dosage, so they tell us that, well, yeah, your vector is too much, or you have the right level of expression. This is really helping us to choose the right dose to start a gene therapy protocol. So that's just one example of a cell autonomous basis of toxicology that we are studying. We can look for a number of synapses, if reduces, apoptosis, things like that. And then in the overall organoid, we're also looking to cellular stress and overall stress to the tissue, not only with drugs, but also with different viral vectors.
Awesome, thank you. I think this question is for Stefan. Was the toxic concentration of your drug comparable in the organoid and in rabbits?
This is one of the standard questions for in vitro models. I think honestly, for the example I have been shown, I don't 100% recall. I think it's never 100% matching. It's always a bit of a different because of the exposure. As said, for the retinal systems, we don't have the barriers present, so the exposure and the disposition of the compound is quite different still in such a model compared to the in vivo. We are in, let's put it that way, we are in a similar ballpark, but I mean, for a very exact interpolation, I guess some of the models, at least, it's still kind of a challenge. But it is an important point, yeah, but it's something we need to be aware of.
Definitely. Somebody asks, do you have any significant issues with limited gas exchange in the center of the organoids causing necrotic cores?
Yeah, definitely is it is a concern. As the organoid grows bigger, the center of the organoid receives less nutrient because it's all about diffusion at that stage, and this is gas and other nutrients. And that's why I think one of the next big steps in this technology is to vascularize the organoid. So my lab, other labs are looking for ways to either induce vascularization within the organoid by generation of endothelial cells that self-organize, or using microfluidics in a very artificial way, but that does the job. I mean, it penetrates the organoids and help on the diffusion. It is a concern, but one thing that we also learn is that it happens to the normal developing brain as well. The normal developing brain press to this phase of hypoxia, which might be a signal for the vascularization to kick off. So this is something that it's fascinating, and as I said, lots of labs working on that. I think that in the next 3-4 years, we'll overcome that.
Awesome, thank you. For Stefan, he said, there's a question, was there anything in particular you did for your addition of microglia to the spheroids to get full integration?
Yeah, that's a good question. It's no, it's for whatever reasons, there seems to be some natural cues that they really like to be integrated. There was no need, at least here from the neurons, the 3D neurons. Yeah, there was no need for any additional supplemental factors. So they feel quite well at home after you put them together.
Great. Also for Stefan, can you comment on the reproducibility of retinal and CNS organoids? Can you quantify? So I guess, can you give a few more details on that reproducibility aspect?
Yeah, this depends heavily on the protocol which is going to be used, right? So the spheroids, as this is a spheroid protocol, it's quite homogeneous because it forms rapidly compared to an organoid protocol, which takes, I mean, retinal organoids takes ages until they get into a, I wouldn't call it a mature stage, but I mean, an advanced stage, which of course brings much more heterogeneity. And then for the retinal organoids, you need to cut them into pieces and dissect them. I mean, we have looked into different ones from also different providers and collaborators and so on. And it really is a multifactorial problem. That's, I mean, one thing we discussed a lot is maybe automation could bring some more homogeneity to this entire field. But as said, for the organoid protocols, it needs also still some manual handling to some extent in the labs, and this brings in some heterogeneity. But the spheroid protocol looks pretty homogeneous.
Thank you. This is for Alysson. So notice the models by the first speaker used iPSC-derived cells. Can primary neuron astrocytes from preclinical species be used for forming spheroids? What is the limitation?
Yeah, they could, but it's not the same. We actually did this experiment side by side. Once you have like preformed neurons and astrocytes and put them together, yeah, you create like a nice way to study the interplay between these cell types. But it's very different from the developing system where you get the cells to interact in a more physiological way. So yeah, I think it depends on the application. For example, if I'm studying like the communication between neurons and astrocytes, this might be okay. But now if you really want to understand how this communication is established during development, or how can you perturb during development, then I think getting from scratch from the iPSCs or human ESCs and create them, it's a much better approach.
Great. This one's for Stefan. How close is IC50 relative to Cmax for known standards and in both models? ?
Yeah, again, the dose / concentration. As said, I mean, I guess we are in a similar ballpark, but it's not exact, right? It's really hard to answer. It really depends, but this.
Yeah, definitely. For Alysson, at what state of the organoid maturation can one detect ictogenic effect of compounds such as seizure phenotype?
Yeah, so interesting. It depends on the condition. For example, we work with CDKL5 deficient syndrome. We see as early as 4 weeks. So this means that it's totally independent of the inhibitory neurons. Just with excitation, you already see hyperexcitability. Other conditions like Rett syndrome, we need to wait a little bit. It seems to be dependent on the inhibitory neurons. The beauty of the protocol is that you can analyze at different time points and have those questions. For example, for some of the conditions, you can block the emergence of GABA by adding GABA inhibitors at different time points and see what happens to see if that cell type is really important or not. But I would say that the earliest that we see something such as hyperexcitability would be 4 months.
Great. Somebody is interested in the cyno IPS neurons and was asking if those are commercially available for Stefan.
As far as I know, I mean, not. The IPS line is something we did many years back, and we just applied it for this experiment. So I'm not aware of a cyno IPS line which is commercially available. Yeah.
And I can also throw that out to any of the developers that may be on the chat, and I'd encourage you to attend the workshops next week where the developers will kind of be presenting their commercially available models. Is it possible to conduct the oscillation evaluation in 2D culture? Do you have any experience?
Yes, people have done that with primary neurons or even tissue isolated from epileptic individuals, biopsies. You can plate on the organoids and see it. You can definitely see oscillations there, but they won't last long. So that's the advantage of the organoid system. Allows you to have like a longitudinal analysis. So you can keep this organoid for several years. The explant or primary cells will last a couple of weeks only. So that's the difference.
Yeah. Another question would be, I think this would mostly be for Stefan, but I think it could actually be for either, is to your knowledge, have any neural models been used to either rule particular drug candidates in or out, support IND enabling studies, etc., or is the industry more in this exploration phase?
I can go first. It depends again. I mean, here it depends on which indication we are talking about. As said, in oncology, for example, the bar is lower. Yeah, we use some of those as part of an IND enabling package, being looking into neurons. For more CNS indications and chronic indications, I guess it would be really hard at the moment, to convince also regulators with an in vitro only package, for example, or as looking only into an in vitro model to bring something into phase 1 clinical studies. But yeah, as said, it depends a little bit on the indication.
Absolutely.
But we use them routinely for some early compound testing and really looking into different tox profiles of different compounds in a similar indication.
Great. Alysson?
I echo what Stefan said. I think it really depends on your question, on the indication. And in some instances, it's a big yes, so it's ready to go. In other instances, it might not be on the prime time yet.
Totally. And this is kind of a similar, but in your experience, how do the regulators view these systems currently when you talk about, presenting them as part of a filing? I don't know if you can comment on that or not.
I'm not there yet, so okay. I will let Stefan answer.
I think using in vitro models in drug development and also submitting them part of an IND, IB is, we are doing that since a long time. I think it depends less on the model system, but rather on the scientific question, how well this is going to be addressed and formulated and how much your data really convincingly supports argument A, B, or C. So I think, having just, also had a roundtable discussion on that this year, I guess the regulators per se are very open receiving such data It really depends again on the question and how this can be addressed. It's not so much a focus on the model system needs to be relevant, but it's more like the overall scientific concept needs to be thought through. And of
Absolutely.
course, you cannot bypass the guidelines by, this is then another thing. But having that as an additional mechanistic readout, I think we do that since many years, and then it really depends.
Yeah, thank you. Is it possible to model different, I mean, I think you kind of went into this, but model different areas of the brain? How do you go about this?
Yeah, absolutely. The first thing you need to know the protocol to create that specific brain region. And I must say that we are at different stages for the different brain regions. I mean, it's much easier to create a cortical organoid than a cerebellar organoid, where the cells are more complex. This is a postnatal part of the brain. So it takes longer, and we don't know all the factors during development that actually leads to that part of the brain. Something for the hippocampus. It's also something that will develop more postnatally. So you are talking about a very long protocol to get to that brain region. So these are some of the technical limitations. But again, I think we'll eventually overcome those. But once we have that, then yeah, it's a matter of developing your protocol, optimizing it, and then you're ready to go. And the other thing that you can do is to see the interaction of that brain region with other regions. But we are still creating formulas for the different brain regions. I said, I mean, the cortex is really good. We have cortical plates, different neurons. If you look at the thalamus, the retina is also really good as well. But the thalamus is highly complex, and in vitro we have something that's very simplistic. So we are not at that level yet. So it really depends.
Absolutely. Any other comments on that, Stefan?
Yeah, we haven't explored that, I mean, for toxicity assessment yet, but yeah, I mean, in literature and what, yeah, it seems to be possible. As said, it's to me is, yeah, for toxicity, it then depends very much on the question if you would need to go down that effort or how much you need to recapitulate this. Ideally, you would love to, but then it depends what's available.
Yeah, absolutely. So I think this might be the last question. Has anyone explored the impact of matrix composition, whether synthetic or animal-derived, on phenotype?
We are doing a little bit of that and testing different matrices, different ways to see how much they impact the organoid, and also to see if the cells are responsive or not in a positive or negative way. And I know other groups are also doing that. So I think that it's an active research in this field.
Great. Awesome. So that's all the questions for today. Do either of you have any final comments or thoughts that you'd like to share with the group before we wrap up?
I would say what I always said to my students, that this is a growing field, very young field, lots of opportunities, room for improvement, both at the protocol and in uses. You can go from plug organoids into robots to grow organoids in microgravity to see the impact of space environment on aging, for example. So there's lots of applications that one can use this field, and I think replacing animals is something that is inevitable. I think we're going to get to that stage.
Absolutely. Stefan?
Yeah, I mean, I totally agree. It's an evolving field. It's massive what has been happened in the past handful of years or so, and also in pharmaceutical industry is kind of an explosion, I would almost say. So yeah, and there's tons of applications and a lot of opportunities. So I think overall the environment is very open to see and support and gain acceptance of these models also for drug development.
Absolutely. Awesome. Well, thank you guys both so much for speaking today. Thank you for everyone who attended, who asked questions, who asked who's participating in the field. I know we have a lot going on. Again, we've got our workshops with our companies on September 21st and 22nd. So we hope many of you can join us for those. Hope you have a wonderful rest of your day. And thank you again. Goodbye, everybody.
Thank you.
Bye.
Bye-bye.
Bye. Thanks.